नमस्कार, हमेशा खुश रहो बिटिया. I am JV'n Dr. Trivedi, and today I am going to take the lab session of B.Sc Agriculture second sem. And the topic for today is measurement of photosynthetic carbon dioxide assimilation by Infra Red Gas Analyser. In short, we call it as IRGA. Now, if we look, what is it? The measurement of gas analyzer, infrared gas analyzer is used to study the rate of carbon dioxide evolution from a leaf, and this leaf is enclosed in a gas chamber, and the gas surrounding the leaf is circulated by a pump to an infrared gas analyzer. This gas analyzer component mainly comprises of a light source which gives off infrared rays which fall on a photoelectric detector. Now, the carbon dioxide released from the leaf absorbing the infrared rays when cross the infrared beam, that is the IR beam, some of the rays are intercepted and the detection response changes. And thus it is possible to measure the amount of carbon dioxide released initially and then after specific periods of darkness or illumination. So this kind of gas analyzer is used for measuring the carbon dioxide release. So it can determine the amount of carbon dioxide release. Now the leaves of photorespiring plants are kept in the chamber, and one end of this chamber is connected to a source of a mixture of isotopes in the form of gaseous form, that is the 18 molecules of O2, and in the other end is a mass spectrometer. The spectrometer instrument is employed for the exact determination of the isotope. I will be sharing with you the diagram of a gas analyzer. So here you can see, as I have already explained to you before, now this is a gas analyzer. This is the tissue of the leaf from which we are going to measure the amount of carbon dioxide evolved. Now this is a living tissue, and we can take the readings at different environmental conditions after different time period of illumination or dark period, and measure the amount of carbon dioxide evolved. Now here you can see, and when we measure the amount of carbon dioxide evolved, that means we can measure the amount of photosynthesis it is going on. So from here, this is a pump. This is the carbon dioxide evolving, and this is the infrared gas analyzer. And from here, it will pass through here, and these are the electrodes which are going to measure, and this is attached recorder. So the amount of carbon dioxide passing through will be following on these photoelectrode detectors, and this will give the reading in the form of the response changes in the recorder. So this is the basic principle of the gas analyzer. So students, I hope you have liked my lecture, and if you have any query, please give in the comment box. I will resolve it soon. This session was powered by digital version 2.0 of Jyoti Vidyapeeth Women's University. Thank you. Namaste.